Welcome, everyone. I'm Kira Curtin, Senior Editor at GenomeWeb and 360DX, and I'll be your moderator today. The title of today's webinar is A Clinical Lab's Journey to Gastrointestinal Syndromic Molecular Testing Using Barcoded Magnetic Bead Technology, and is sponsored by Applied BioCode. Our speaker today is Kendra Pesco, the Technical Director of Infectious Diseases at TriCore Reference Laboratories. You may type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar presentation. If you look to the bottom tray of your window, there are a series of widgets to enhance your webinar experience. And with that, I'll turn it over to Kendra. Kendra?
Great, thank you, Kiara. Trying to progress the slides. Okay. So I'll take you through my journey into using this molecular diagnostic MDX 3000 system. This is just an outline of what we'll be discussing today. So I'll describe the pathogens of interest and our previous processes for testing for them here at TriCore. I'll detail the Applied BioCode workflow, how the instrument works, and the approach that we use with this testing at TriCore. I'll take you through some of our validation results and some current metrics for performance, and talk about a few of the future directions. So what is syndromic testing for GI illness? Well, the symptoms of GI illness are fairly common. They've probably been experienced by everyone that's listening to this talk. They include diarrhea, most importantly, abdominal pain, stomach cramps, nausea, vomiting, and really the risk from GI illness is dehydration. There's numerous causes for GI illness, including infectious causes from bacteria, and here we'll focus primarily on the parasites and viruses. But there's many non-infectious causes, including intolerance for a certain food, celiac disease, irritable bowel syndrome, and pregnancy, just to name a few. So there's many, many causes that can be non-infectious. So some of the primary infectious agents that cause acute GI illness are bacteria, including Salmonella, Campylobacter, Shigella, E. coli, various kinds of E. coli, and C. diff, which causes primarily an previous antibiotic treatment-associated diarrhea. Viral diarrhea is caused by caliciviruses, of which norovirus is the most common, rotavirus A, adenovirus types 40 and 41, and astrovirus. The protozoa that primarily cause GI illness here in the U.S. are Cryptosporidium and Giardia. We do have seasonal outbreaks associated with Cyclospora and Entamoeba histolytica. So I'll talk about Cryptosporidium species first. So cryptosporidiosis, most outbreaks of cryptosporidiosis in the U.S. are related to pools, water parks, splash pads, other shared water areas. So a really important point here is to not swim when you have diarrhea. And actually for 2 weeks after diarrhea resolves, it's a good idea not to get into these public areas because it's very easy to pollute both pools and water parks with Cryptosporidium. Some outbreaks also come from cattle, in contact with cattle, or drinking of milk. And then childcare. Childcare is actually going to be a common risk factor throughout. So anything with fecal oral transmission, which is what most of these pathogens, which all of these pathogens have actually, is fecal oral transmission. And so anything with fecal and oral transmission is going to be prevalent in childcare settings. So this is how we look, one way that we look for Cryptosporidium. And the bottom left side, we have a wet mount. And these two pink arrows are pointing to Cryptosporidium cysts. They're 4 to 6 microns across. The brown arrow here is pointing out a budding yeast. We also stain stool specimens with trichrome to help pathogens, especially protozoan pathogens, stand out. So these red arrows are also pointing to Cryptosporidium cysts. Another stain that is commonly used is a modified acid-fast stain. These Cryptosporidium oocysts appear 4 to 5.5 microns in diameter, and you can sometimes see sporozoites inside the cysts. So the first described water-associated outbreak of Cryptosporidium here in New Mexico was associated with a pool, the West Mesa Aquatic Center. This occurred in 2008. It started with recognition of 6 children from 2 families that had diarrhea, vomiting, and fever after having swum at a pool on August 2nd. After that, there was a confirmed case 1 day later, and a 17-year-old competitive swimmer who had been experiencing diarrhea since mid-July continued to practice and compete. On August 15th, samples from all 3 pools at this West Mesa Aquatic Center were sent to the CDC, and all 3 tested positive for Cryptosporidium. Subsequently, through public health efforts, there were ID'd 29 confirmed and 60 probable outbreak-related cases. Of these, 30 had reported recreational activities while ill. So how do you solve this? Well, hyperchlorination has to occur at the pools and any water park where these infected people swam or played. And when you hyperchlorinate, you actually have to shut down these recreational facilities. So it does lead to some economic impacts for these facilities. And actually, the city of Albuquerque changed the way they were treating their pools after this occurred. So this is one reason why Cryptosporidium spreads so readily in pool or water park settings. E. coli can be destroyed within 1 minute, or at least 99.9% reduction in population within a minute of contact with chlorine in water at 1 milligram per liter. Hepatitis virus lasts more like 16 minutes. Giardia is a little bit hardier at 45 minutes, but Cryptosporidium can actually last 10.5 days. So it's just extremely hardy and resistant to disinfection with chlorine. So Giardia lamblia is the next species that I'll speak about. Again, it has fecal-oral transmission. Transmission occurs when you swallow unsafe food or water that's been contaminated with Giardia. Again, this occurs in childcare settings, but it actually occurs frequently in people that are camping or hiking and might consume water that has not been treated. Typically, symptoms begin 1 to 2 weeks after infection, and you can be infected and asymptomatic. Actually, all 3 of the protozoans that I'll speak about today have some population that when they're infected, they are asymptomatic. Most mammals can actually carry and transmit Giardia, and that includes cattle, beaver. It's known to campers as beaver fever. So this is an example of a cyst in a wet mount. Here's a picture of a trophozoite, so they are flagellated, stained with iodine. And then these are pictures of cysts stained with TriCore. So giardiasis outbreaks in the United States, it's actually the most common cause of intestinal parasite infections. This top map is showing the location of outbreaks in 2017, or from 2012 to 2017 in the United States. And you can see that it's really distributed throughout the United States. This looked at what kind of outbreaks or what the outbreaks were related to. And so many of these are waterborne outbreaks, but there's also some person-to-person transmission, some foodborne transmission, and some that are indeterminate or unknown. The graphs down here show the percentage of the population that tested positive for this across different age groups. So you can see there is a spike in the age 1 to 4. Again, childcare-associated illness. And then this shows monthly rates of infection. And you can see there's definitely an increase of this illness in the summer. Entamoeba histolytica is the last protozoan organism that I'll talk about. We identified this in our, well, we had identified this in our lab primarily by OMP, so an ovum parasite exam. And we would confine this to patients with either a certain travel history, recent immigrants, or people with a compromised immune status. So the OMP exam is actually very slow. It can be manual and somewhat subjective. It's recommended to examine 3 different stool specimens taken over the course of several days. And so you can tell this process is pretty involved. For Entamoeba histolytica, one problem is that you can't morphologically discriminate between Entamoeba histolytica and a non-pathogenic form called Entamoeba dispar. This is a wet mount showing what Entamoeba histolytica looks like. So it exists in the human body both in a cyst form and a trophozoite. Typically, when people excrete the cysts, it'll be in a more solid stool. The trophozoites might come out in a bloody diarrhea, and bloody diarrhea is a characteristic of infection with Entamoeba histolytica. You can see this diagram shows red blood cells that have been ingested. That's one way to determine whether it's histolytica or dispar, but dispar has been known to ingest red blood cells as well. And the problem with amoebiasis, or infection with Entamoeba histolytica, is that not only does it replicate in the gut, it can actually disseminate to other tissues. So extraintestinal disease does occur, including encystment and forming of an abscess in the liver. Also, sometimes the lungs or the brain can end up with abscesses from this organism. So it's very dangerous. So treatments for parasitic GI illnesses actually exist, and they work fairly well. So if you can determine that the illness is caused by a parasite, treatment can occur. So nitazoxanide is the treatment of choice for Cryptosporidium, and that has a cure rate of around 80% for immune-competent patients. If you have a compromised immune system, you may need treatment for a longer period of time or a different treatment. People with compromised immune systems actually can be affected very poorly by Cryptosporidium. Giardia can be treated with metronidazole, tinidazole, or nitazoxanide, and Entamoeba histolytica is typically treated with metronidazole or tinidazole. So what are the viral pathogens that cause GI distress? The ones of interest here include norovirus, G1, G2. This is the leading cause of vomiting and diarrhea in adults in the United States, and symptoms typically occur for 1 to 3 days. Rotavirus A is a vaccine-preventable virus, which occurs in young children, typically occurs in the winter, as does norovirus, and symptoms typically occur for 3 to 8 days. And another GI illness causing virus is Adenovirus 40 and Adenovirus 41. So norovirus G1, G2 is the most common cause of GI illness in adults in the United States. It causes around 900 deaths per year, 109,000 hospitalizations, 465,000 emergency department visits, and 19 to 21 million total illnesses. This is showing the average number of reported norovirus outbreaks by month from 2009 to 2017. You can see there is some foodborne outbreaks that occur, but most of the transmission is non-foodborne, so person to person. And the majority of outbreaks are caused by G2. G2 is the darker bar here versus G1. This is data from the past 2 years. There's a program at CDC called CaliciNet that actually tracks outbreaks of norovirus and genotypes them over time. So you can see there's a very pronounced winter peak for norovirus, and G2 is the genotype that's most closely associated with outbreaks. So rotavirus A, rota means wheel, and you can kind of see that wheel shape here in these slides. Again, fecal-oral route of transmission. It's a double-stranded RNA virus, and it used to be the most common cause of diarrheal illness, with almost all children infected by their 5th birthdays. Symptoms include vomiting, diarrhea, fever, abdominal pain, and most worrying, dehydration. And it actually still causes quite a bit of illness. Only around 70 to 75% of children are vaccinated for this pathogen. So we still see a winter peak of illness for this pathogen. And then adenovirus 4041. So there's many different strains of adenovirus that can infect people, but 40 and 41 are primarily associated with GI illness. This causes gastrointestinal illness primarily in young children or immune-compromised populations. It can be shed for a very long period of time, and it typically causes symptoms that last 5 to 12 days. It's a DNA virus. Some of you might be familiar with this Health Alert Network memo that came out in April recommending adenovirus testing and reporting for children with acute hepatitis. So there were some children identified with acute hepatitis earlier this year and towards the end of last year that also tested positive for adenovirus 40 and 41. So testing for GI viruses at our lab, prior to the use of the applied BioCode MDX, involved a rotavirus EIA, so this immunocard stat test, which involved manual resulting and relatively poor sensitivity. And we had to test a norovirus send-out test that we still actually offer. But that has a poor turnaround time. So most of the time when you use a send-out test, it takes around 7 days to return results. Testing for GI parasites that occurred here before adopting the applied BioCode, we used OMP examination. Again, OMP, ovum parasite examination, is microscopy that occurs from raw or preserved stool, either in a wet mount or a stain slide. We used stain slides with trichrome. You can concentrate stools to improve sensitivity because sometimes very few parasites are excreted in each stool. And the CDC actually recommends 3 stool samples collected on different days for that reason, because few parasite cysts might be excreted in each stool. The OMP exam is subjective, it's high complexity, and you have to have really highly trained personnel to see these organisms. And then it's very physically difficult because you're staring into a microscope all day long. So at our lab, prior to adopting the molecular strategy, we actually reflexed orders without travel history or immune status to an EIA for Cryptosporidium and Giardia, which are the most common endemic parasites here in the United States. Unfortunately, we automated that process using the DSX, and it had some issues in terms of pipetting stool samples. So anyone that's worked with stool samples knows that they're very heterogeneous. There tend to be chunks, and so when you're relying on an instrument to pipette from the stool sample, it can be very difficult. So the applied BioCode gastrointestinal pathogen panel is able to detect 17 different targets. It's used on both unpreserved stool or stool and carry Blair. And the targets that it can detect include all of these bacteria, plus the viruses that we just discussed, and the parasite we just discussed. And we adopted this strategy primarily for the virus and parasite testing. So the workflow for the GI panel is an automated DNA or RNA extraction, followed by PCR setup, and then automated PCR detection and results reporting. Prior to loading your extraction platform, you actually have to process the stool. So as I said before, this pathogen panel can be tested on both stool and carry Blair or unpreserved stool. Essentially, you transfer an aliquot of the stool into a homogenizing tube that has a number of different sized beads. And that tube includes a mil of STAR buffer, so stool transport and recovery buffer, and 10 microliters of the RNA internal control used by this platform, which is MS2 bacteriophage. So you transfer either 100 microliters if it's very liquidic, which doesn't happen too often, or you can use a loopable. You vortex on medium-high for 5 minutes. So we use this nice foam insert on our vortexer. And then centrifuge for 2 minutes at 3,500 RPM. Extraction can occur on 2 platforms, and we actually validated, and have used clinically both platforms. The EasyMag was the first platform that we brought on. This platform's fairly nice, except that you have to come back 10 minutes into your extraction and add magnetic beads to all of the samples. So it kind of doesn't give you as much downtime in between loading the instrument and unloading the extracts. You also are limited to 24 samples at a time, and I believe this instrument is being sunsetted by bioMérieux. We've more recently begun to use the MagnaPure 96 from Roche. This allows extraction simultaneously of up to 96 samples, and it takes around 50 minutes to perform that extraction. Once your extraction has been completed, you might set up your PCR plate. We use the master mix that this platform uses is just 3 components, so it's fairly easy to set up. Master Mix A, this GPP primer mix, and an RT mix. You load the master mix into a 96-well plate, and then you load your extracted samples into that plate. And then you load the plate and reagents and other supplies onto the MDX 3000 platform. This is the MDX 3000 platform. It's around 61 inches high, 36 inches wide, well, 42 inches wide and 36 inches deep. It has attached a monitor that walks you through the setup of the instrument and a barcode scanner for scanning in both reagents and supplies and your sample IDs if need be. You can see there are tips loaded onto this platform, as well as a hybridization plate. There's a thermal cycling unit in the back here. And these doors actually give way to a little storage area. So this instrument works by performing a reverse transcription, then a PCR, and 1 primer for each of the pathogen targets is 5' biotinylid. So you amplify denature, and then your probe that's 5' you add a probe that will detect the amplicon that is attached, that becomes attached to a barcoded magnetic bead. That barcoded magnetic beads get mixed with the PCR amplicons, and they attach to the biotin labeled amplicons. Unbound primers are washed away, and then the streptavidin phycoerythrin gets added into the plate. And that brings the phycoerythrin into proximity with barcoded magnetic beads. Unbound SAPE gets washed away, and the phycoerythrin is then excited. And they use a microscope to actually overlay the bright field and fluorescence images. And so the fluorescence provides an idea of the power of the signal, and the barcoded magnetic beads can be read by the microscope and gives the identity of the pathogen present. So what is a barcoded magnetic bead? It's a fabricated digital barcode, just like you would see at the supermarket, or you might scan in with your container ID. So essentially, the barcode scanner is similar to the microscopic scanner. It's just a much, much, much smaller barcode. So we performed a validation of this platform in 2019 to 2020. We looked at Crypto and Giardia, and we compared them against our Crypto Giardia EIA with the DSX automation. We compared Entamoeba histolytica against the OMP results, and Rotavirus A against the Meridian Immunocard stat results. We performed an evaluation with the BD-Max enteric viral panel and enteric parasite panel at the same time, so we used that to resolve our discordance. So the BD Max is a smaller platform that uses this cartridge-based extraction and then loads the extract onto a card for PCR to occur. We had previously brought on the BD Max for bacterial targets and currently use it to test for Salmonella, Shigella, Campylobacter, and Shiga toxin-producing organisms. So here are some of our results. So our validation results for Cryptosporidium looked really good. We found 100% agreement between the two methods. Our positives were all positive by both methods, and the negatives were negative by both methods. So we found that really promising. Our results for Giardia were a little bit different. So we saw quite a few positives for Giardia during this time period, but then many that tested positive by our EIA and were negative by the BioCode. Subsequent testing on the BD Max showed these to be true negatives, and we concluded that it was false positive on our reference method. There was one sample that tested negative on applied BioCode negative on a reference method and positive by applied BioCode. And this was actually a true positive. It was positive by BD Max, and we saw it testing positive when we repeated it on applied BioCode. So the overall sensitivity for this platform is 100%, specificity 98.3%, and agreement 98.6%. For Entamoeba histolytica, we saw 5 that were positive by both methods, and 1 that was positive by our OMP exam, but negative by applied BioCode. Unfortunately, we did not have an extra aliquot of this stool, so we were not able to resolve this with BD Max testing. We assume this may have been a dispar rather than a histolytica because the GI panel from applied BioCode will only detect histolytica, and there was no way to discriminate between the two on the OMP exam. But because we do not know, we have to put our sensitivity at 83.3%, specificity at 100%, and agreement at 98.1%. For rotavirus A, we saw somewhat similar results to Giardia in that we had fairly good agreement across both the platforms. There were 2 that were detected as positive by a reference method, but negative by applied BioCode. These also were negative by BD Max. There was 1 that was negative by a reference method, positive by applied BioCode, and this was also positive by BD Max. So from this, after discordant resolution, we estimate our sensitivity and specificity at 100%, and we saw 100%. So this diagram gives an idea of the overall workflow and time to results for the GI pathogen panel. So prior to setting up the platform for 96 samples, you have to do a bead lysis and extraction. Once the extraction platform is set up, you have this walk-away time where you might perform a different assay or use it in a different manner for around 70 minutes. The PCR setup takes around 30 minutes, followed by 10 minutes to set up the instrument itself. And once the instrument is set up, if you're running a full plate of 96 samples, it will take 3.5 hours. Here at TriCore, we did adopt the applied BioCode instrument for testing of viral and parasite samples. We run this as batch testing that we were running every other day, and actually just recently we've moved to an everyday batch testing for these organisms. The benefits of adopting this include interfaced resulting, and we have one workflow for 2 different orderables. And then the smaller panels, by dividing it into both a viral panel and a parasite panel, really allow providers to order based on need, so maybe season, the symptoms the patients are presenting with. So those viral pathogens tend to be a more aggressive, quicker onset of sickness and quicker resolution as well. Whereas some of the the parasites tend to cause diarrhea that might last for a longer duration of time. And then, of course, that seasonal difference between the viruses that tend to peak in the winter and the parasites that peak in the summer. So we have these as two separate ordables that are performed on the same platform. This is what the report looks like. So they're able to mask any of the non-ordered targets. So you can see Campylobacter, Clostridium, and these are the different samples that have been loaded. And A, because that's not what was ordered. In this instance, the GI parasite panel was ordered, and so this one was negative for Crypto, E. histolytica, and Giardia. In this instance, the viral panel was ordered. This sample was negative for Adenovirus, Norovirus, and Rotavirus. We include a negative extraction control with all of our runs and a positive extraction control that includes one of the pathogens being tested. And this is an example of an unmasked result for a single patient. So you can go back into the system and unmask results if needed for further investigation, or I'm going to show some results from a retrospective analysis that we performed. So we also did a retrospective evaluation looking at norovirus and comparing it to our send-out PCR, which is performed for genotyping over October 2020 to September 2021. And I'll also show some results from a retrospective analysis that we did this last fall and winter looking at bacterial targets. This will be compared to BD max, and then all of our Campy, Shigella, Salmonella positive stools actually get sent out to the New Mexico State Scientific Labs, where they perform isolation and some genetic analyses to see if they're related. And that helps to identify outbreaks for these bacterial pathogens. But we were able to use the results of culture to resolve any discordants that were positive on BD max but negative by applied BioCode. So for norovirus, we did a comparison looking at almost a year of data. We found there were 66 positives in agreement. There were 5 that were positive by the reference method but negative by applied BioCode, 1 positive by applied BioCode and negative by the reference method, and 354 that were negative by both tests. So the overall sensitivity was 92.9%, specificity 99.7%, and agreement was 98.6%. For Shigella, we found fairly good concordance across the platform. There was 1 that was negative on applied BioCode and positive by BD max. This had no growth in culture, so we determined this to be a true negative. Of course, that could be caused by a difference in the limit of detection between the 2 assays as well. And you would expect that if a sample has a very low concentration of pathogen, it might not culture as well. And then 1 that was positive on applied BioCode but negative by BD max. So overall sensitivity for Shigella was 100%, specificity 98.6%, and agreement 99%. For Campylobacter species, we saw a very different pattern. So we found 18 that were positive by both methods, and 22 that were negative by applied BioCode but positive by the reference method. Looking into these a little bit further, we saw that they actually had very low CT values on our reference method, the BD max. And looking at the results given to us by the state lab, 19 of 22 of these were actually able to be cultured by the state lab and confirmed to be Campylobacter jejuni. So with this data, we estimated sensitivity for Campylobacter at 45%, specificity at 100%, and one fortunate thing about working with applied BioCode is that they're very proactive in supporting the work that we perform. So Fortunately, we were able to capture 3 discordant stools and send them to the Applied BioCode team. They tested these with another lot of reagents and confirmed detection of Campylobacter. They tested them after dilution with the current lot and also were able to detect Campylobacter. So they determined this to be a lot issue for the GPP reagents that we were using. We did a subsequent analysis with a new lot of reagents once one was received in April and May of 2022, and concordance for Campylobacter detection with this other lot of reagents was 16 out of 16. The comparison for Salmonella species is shown here. It's fairly similar to the other comparisons except for Campylobacter in that there was fairly good concordance between the methods. There were 3 that tested negative on Applied BioCode and positive on BD Max. 2 of these had no growth in culture, and we determined them to be true negatives based on that. And 1 grew in culture, so it was a false negative by Applied BioCode. Overall sensitivity for Salmonella is estimated at 95.7%, specificity at 100%, and agreement at 99%. And for STEC, we saw a similar pattern where there was fairly good positive concordance. There were 2 negative on Applied BioCode that were positive on the reference method, but these also had no growth in culture, so we determined them to be true negatives. And there was 1 that was positive on Applied BioCode, but negative on the reference method. So given these data, we calculated sensitivity at 100%, specificity at 98.9%, and agreement at 99%. So another thing that we look at with stool testing is what is the overall invalid rate? We certainly see that stool can impact testing in that it inhibits PCR. It also can, as mentioned earlier, clog liquid handling machines and lead to problems with liquid handling machines. This is giving the invalid rate based on testing from January to July of 2021. Looking at over 3,000 samples tested, around 370 were invalid on the first run. There is a reflex for stools that are invalid initially, where you do a slight dilution into star buffer described in the package insert. So we do follow this pattern if we see any invalids. So on repeat, 101 of these were invalid on the second run. So that gives an overall invalid rate of 3.17%. And of course, we don't charge for these, we credit the test, and it's an unfortunate aspect of testing stool that some of them will test invalid. So what are the benefits of our overall switch to molecular GI testing? We believe we're able to provide more accurate test results to providers. We can decrease our turnaround time overall, especially with daily batch testing using this platform. We interfaced our reporting, so that minimizes transcription errors, and it also takes less time to result out tests. When we do have issues with the platform, they've been very quickly addressed by the Applied BioCode team, so we are very happy with their troubleshooting and support that they provide. And then the workflow is easier to train than O&P exam and easier to coordinate with new technologists. So what are our future directions with this platform? We have discussed using it for additional bacterial targets. We've looked at possibly validating as an LDT to report norovirus genotype, since many providers and especially infection control people are still interested in seeing the genotype for norovirus and still utilize our send-out test for that. We've considered offering a large GI panel, so including all of the targets in a panel, so that providers can have that full coverage, and potentially utilizing other LDTs on the Applied BioCode platform. So that's all I have for you today, and I'm happy to take any questions.
Thank you, Kendra. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. We'd like to ask attendees to take a moment after the webinar has ended to take our exit survey to give us your feedback. And so now we'll start the Q&A portion of the webinar. And the first question is, what is the value of using syndromic panel versus individual targets?
So there aren't FDA cleared tests for each of those individual targets. And if you have LDTs for each of those targets, it actually has a lot of evaluation and paperwork and difficulty in the background. So if we were going to use a separate LDT for each of those tests, it might also be a different workflow. So that's more training for your staff. It's I think it can be a struggle to implement in the lab. It also makes sense to offer a panel that tests for the pathogens that could be causing one pretty consistent symptom, which is diarrhea. So there's so many different pathogens that can cause that really using a panel kind of allows you to test for all of them simultaneously.
And how important is it for your lab to be able to perform a test that can differentiate between Shigella 1 and 2 E. coli?
I don't know that differentiation between Shigella 1 and 2 is a very important issue in our lab. I think identifying that Shigella 1 or 2 is present is what's important in our lab.
And clinically speaking, does treatment or patient management vary between norovirus G1 and 2?
I don't believe so, but I think in an outbreak setting, you do sometimes want to determine whether it's G1 or G2 and whether the isolates, that can help you determine whether the isolates or the positive stools are related to one another. So is it an outbreak or is it just, winter vomiting sickness? Lots of people have it and might have been from two separate sources. So I think it's important from an infection control aspect, but not as much clinically.
Why do you only use the BioCode for viruses and parasites and not bacteria?
We have another platform that we use for the bacterial testing currently.
Okay. And so does a cyst or trophozoite form affect the diagnostics? Is either form dominant in stool?
Well, the cysts are dominant in stool most of the time, especially for Cryptosporidium and Giardia. That's what you typically look for in the OMP examination for Cryptosporidium. But I believe this test actually, because it's looking at nucleic acid, should be able to detect both.
Do you use the masking option provided on the MDX 3000? And, if so, how is that done? And could the results be unmasked after the run is complete?
Yes, we do. So I did have some slides that addressed that. So we do mask all of the targets that are not ordered. So again, here you can see the Crypto, EHIS, and Giardia have been ordered for this patient, the internal control value. And then for this patient, it's the GI viral panel that was ordered. So you can see the Adenovirus 4041, Norovirus G1, G2, and Rotavirus A as the pathogens that are being detected. I think it's very important to have that masking available. We wouldn't want our technologists to see Campylobacter species detected and, then run out and feel like they need to do something with that result. So it's very helpful having that masking available. You can go back and retrospectively unmask results, and we did do that when we were performing the retrospective evaluation for the bacterial targets. It can be helpful for other quality projects or validations in the lab as well.
When you performed culture on discordant results and you got a negative culture, you seemed to consider that sample was a true negative. How did you take into consideration the possibility that the microorganisms could be inactivated or quote unquote dead?
Sure. I mean, that's a very good comment on it. So the microorganisms could be inactivated or killed, certainly by the time the culture took place. They also could be at a level that's just too low to detect by culture. So there are some caveats to considering those true negatives. I think it was an option that we had available, and so we used it in order to look at those discordances. But it wasn't perfect in any way.
And does your lab use the user-defined mode and develop LDTs?
We do not currently, but we are interested in looking at using that mode.
And what is the dilution for reflux testing?
Ooh, I believe it's like 100. I'd have to look it up. It's in the package insert,
okay.
And perhaps someone from the Applied BioCode team could speak to it. I think it's something like 100 to 200 or 50 to 150. It actually can happen in the extraction plate, so it's fairly easy to use. It's a fairly minor dilution. It's no more than 1 to 5.
Okay. And does the BioCode use a lot of consumables per run?
It does not. I can actually bring up the picture of it, and maybe we can look at some of the components that get loaded onto the instrument. So this is the picture again. Hopefully you can all see it. So there are 2 racks of tips in here, and it actually uses by column. So if you have 3 columns of testing, it'll use 3 columns of the tips in the back. For the larger tips, it uses 3 columns no matter what. Those are used to actually distribute your barcoded magnetic beads and your strep avid and phycoerythrin to the hybridization plate after the thermal cycling has occurred. It uses 1 PCR plate, 1 hybridization plate, and a hybridization lid for each reaction, and then strep avid and phycoerythrin aliquot. So overall, the amount of waste that occurs from this instrument is fairly low.
And since you have a BD Max, what was your motivation to use the Applied BioCode method?
When we initially performed the evaluation, there were some issues with false dual positives on the BD Max. And so we felt like the accuracy on the Applied BioCode was better.
And let's see, sorry. And did your lab validate other extraction methods for use with the MDX 3000? If you did, kind of like, sorry.
No, it's fine. We didn't validate any off-label extraction platforms, but we did validate both of the extraction platforms that are on label, so the MagnaPure 96 and the EasyMag.
And what is the maintenance of the Applied BioCode like? How has that been?
It's been pretty good. There's just a once weekly water wash, so it takes around 10 to 15 minutes, but the actual prep time to perform that is around 5 minutes. There are PMs that occur every 6 months, just like most platforms. So it's relatively low maintenance.
And does stool processing for 96 samples increase the processing time by a lot? About how much time would you need? Yeah.
Yeah, so you're going to have to pipette from each of those samples into an extraction plate. So if you can picture pipetting from 12 samples versus 94, it's going to take quite a bit longer. You're unscrewing a lid, pipetting, screwing the lid back on. If you contaminate your gloves, obviously changing those out, right? So there are aspects of setting up a full plate that make it much more difficult than just, 12 or 15 samples at a time. And it is more time consuming. So I would say it would take around 45 minutes to set up versus 10 or 15 minutes for a smaller batch.
And for solid stool, is it the 10 microliter inoculation loop that you use?
I believe so, but I could be wrong on that. I haven't actually performed a run on this for a couple of years since the validation concluded.
see, sorry, scrolling to see if there's other questions here. Let's see. Yes, and so I guess kind of what were the winning attributes in the end? Was it like flexibility, cost efficiency, having the right targets in the panel? What kind of swayed you?
I think staying the overall performance relative to the EIAs we were currently using spurred us to make the move. The workflow overall is fairly easy, and then the interface resulting is very helpful. So yeah, those three things spurred us.
What do you use for dilution?
So it's diluted into star buffer, and I believe it's 50 microliters from the star aliquot that gets added to 150 microliters of star buffer plus internal control. And that's when it's tested invalid the first time. So when it tests invalid on the first run, our assumption is that the initial loading of that tube, the star buffer tube that contains the beads and homogenizes the stool, that initial inoculation was just too much. And if anyone's worked with stool, like too much is a big problem in terms of PCR inhibition.
Mm-hmm. Well, that looks like all the questions that we've had come in. So I'd like to take this time to thank you, Kendra Pesco, as well as our sponsor, Applied BioCode. As a reminder, please look out for a survey after you log out so you can provide your feedback. And if you missed any part of this webinar or would like to listen to it again, an archived version will be emailed to all attendees. And thank you for joining us for this 360DX webinar.
Great, thank you.